I started it off once, and then it kind of faded, and then I started it a second time. And it's prospered since then. And that's largely because of Christine McNamee, who's a manager, who's been working very hard to make sure that this works very well. And the important thing about the UK Pharmacogenetics Stratified Medicine Network is that we hope this is also part of the landscape for stratified medicine in the UK. Because what it does is brings people together. It brings people together from different stakeholder groups, academia, clinicians, regulators, industry, etc. And we've had some fantastic workshops this year, and we are particularly glad to be co-hosting a workshop with NIHR CRN this time. But we have 3 weeks ago we had a workshop with the Academy of Medical Sciences. We've had a workshop with NHS England earlier this year, and we had a workshop with MHRA this year. And what we're trying to do is to try to discuss the key issues which are facing stratified medicine, personalized medicine, precision medicine, whatever you want to call it, and try to think of potential ways and solutions of trying to solve those issues. What we do is we have a website which highlights all these particular workshops we've had together with the talks. So all these talks today are being recorded and will be available on the website. And the workshops we've had previously are also on the website. And we also have an open meeting as well. And so we had an open meeting with 250 people last year. We've now gone to a bigger venue. I did promise Wembley, but unfortunately I can't afford Wembley. So we've gone to slightly higher, which is about 300 capacity for next year. And there's a banner in the coffee room which tells you when exactly the next open meeting is. And we've got a fantastic list of speakers already for the open meeting, and they're all agreed when we asked them to do it, which means the network is working. So I'm going to sorry about that. I thought I'd start off with that introduction. I've been asked to give you a perspective on challenges of delivering stratified medicine studies. And this is largely based on a document that was produced by the NIHR CRN by Jen Beatty and Nancy Lester. And I'm going to sort of look at those points, but also introduce some vignettes in there based on my sort of experience of what goes on in difficulties in terms of recruiting stratified medicine studies. So the key issue with stratified medicine, as I hope you now know, of precision medicine, is that you do not treat the whole population, you treat targeted groups of patients. And that's crucial if we are going to get particular ways of stratified medicine working and really improving outcomes, hopefully in the whole population eventually when we stratify the whole population, but initially in a small proportion of individuals that are screened. In order to do that, you may have to screen 100% of patients to be able to get 3% of patients, for example, when you have a biomarker which is only present in 3 out of 100. So that means that what happens is that you end up with 3 people you can recruit and 97 you can't, but you'll have done work on 100 people to screen them. And clearly at the moment the NIHR CRN model really relies on total numbers rather than numbers screened, and that might be a disincentive for centers to take part in stratified medicine studies when my R&D department or any other R&D department says, we need to recruit more and more, so we're recognized by NIHR CRN, but they don't really take into account the fact that you have to screen so many to get the 3 patients out of 100. And I guess one of the things hopefully we can discuss as part of the groups this afternoon is what is the finance model exactly at the moment. It's not completely activity based, but how can the finance model be perhaps adapted to take into account the challenges associated with precision medicine. When you are looking at precision medicine and so on, and you're looking at groups of, particular groups of patients, they often are being treated at multiple sites, are being treated across boundaries as well. And again, if you've got a patient in a specialist center plus a district general hospital and so on, and they're going between two different regions, crossing boundaries, again, that can create problems in terms of recruiting, in terms of how this patient is counted in terms of numbers. And again, that is something that would be good to think about, and how we can try to overcome that. So again, as the third bullet point is again related to where the activity starts, where the activity goes to, and where is it actively counted. Again, it relates to the second point as well. One of the issues that I'm very keen on with stratified medicine, and it doesn't really matter how clever you are with your omics technologies, how fantastic you are with your imaging, etc., unless you get your phenotype right, it's not going to work. And one of the key issues is to have a very good workforce who's been trained to really deeply phenotype your patients, clinically phenotype your patients. And that deep phenotyping means that you may be getting a lot of data, and they may require highly trained nurses to really go and do something like that for you to be able to recruit that patient and get it into your CRF. And that enhancement of the phenotype and assessment and so on is going to be crucial in some of the successes that we require with regard to precision medicine. So if you have a workforce that you actually trained and who know how to do that deep phenotyping, but then you have to recruit from a center that's 100 miles away because you're looking at a rare disease, then you can rely on the nurses locally to do this, or you can actually send your own staff who've been trained to be able to do that. And if you send your own staff to be able to do that, they often don't have permissions to actually go into that center. So you then have to get the permissions for them to go into that center, etc., to be able to recruit that patient. And that creates problems as well in terms of time to recruitment. And again, those are kind of practical things that we need to think about. I guess one of the issues is actually streamlining research management and governance. And clearly, some of the things which happen in this country have been fantastic in terms of improvement, and the slide that Nick showed in terms of time to approval and how that's improved is all positive. And clearly, with the HRA coming online and ramping up the activity, hopefully that is going to improve. But what I want to do is to maybe show you a case study of where we particularly struggled and how long it took us to be able to undertake recruitment in one particular area. And this really relates to using health informatics databases and so on. ABPI, in the report said that it's important that we use information technology, which has patient details in there, but we link into biological databases as well. And clearly the FAR Institute, etc., is trying to do all of that and so on. And so what we wanted to do was to utilize CPRD. And CPRD, is a fantastic database that the UK has, which is held by the MHRA. But CPRD has got great health informatics data, prescription data, disease data, but didn't really have any kind of biological samples associated with it. So we undertook some work to really try to link biological samples to a particular phenotype. And what we decided to do was to choose what we thought was a relatively easy phenotype called statin myopathy. There's been a lot of controversy about statins, and it does cause muscle pain sometimes, and we wanted to see how many patients we could recruit through that, because it is quite difficult to identify them routinely through NHS records. So we tried to do that, for example, in the Royal Liverpool Hospital where I work, and we said to the clinical chemistry department, how many CKs are you getting elevated? And they get hundreds of every day, but the majority of those are patients who've been lying on the floor, elderly, who've got muscle damage because they've been lying on the floor, and not really statin related. And we really needed some clear statin related cases of myopathy, and we thought that CPRD would be a good choice of being able to do that. So when we did the study, there were 641,000 patients prescribed a statin on CPRD, and 127,000 of those had concurrent CPK measurements. So we thought, well, actually, we should be able to find somebody here who's got a high CPK related to statin myopathy. So we went about with the governance. So we got one sponsor, University of Liverpool. We got one ethics committee approval for the whole country. And then you had to go to the MHRA and go to the Independent Scientific Advice Committee and get approval. And that happened pretty quickly. But then we had to go to every R&D department within where each GP practice was based. So we had to do 132 approvals for getting single patients from each particular practice. And when we started, my hair was black. You can see what color it is now. each approval was 55 pages long, different questions, and people wanted to change it. They wanted to change the patient information leaflet, etc. And the timelines for approval varied. And you can see what the differences were here. You can see some of them were very good, giving us approval very quickly, but some of them took over 250 to 300 days in giving us approvals. That's after 132 R&D approvals. But we persevered, and Anita, who's here, who works with me, was particularly keen on persevering, and so on, and we managed to recruit patients. And this shows you the flow diagram in terms of the difficulties in recruiting and the attrition rate you get in terms of recruiting. So if we consider there were 640,000 individuals, and CPK was measured in 127,000, our phenotype was CPK above 4 times upper limit of normal, which got it down to only 712. So we went from 640,000 to 712 individuals. But then when you actually start contacting the GPs, you get a high attrition rate as well. And eventually we ended up with adequate samples in about 115 patients from that initial 712 individuals that we identified with the right phenotype. So there's high attrition rate despite using these databases and so on. And those challenges, and I completely agree with in terms of the fact that these health informatics databases are going to be crucial in terms of stratified medicine, but in terms of then recruiting them to particular studies and doing these kind of biological studies, there are going to be issues, and there is going to be a high attrition rate in terms of taking forward precision medicine. And there was a 1-to-10 recruitment ratio, as you can see on the slide. And you can see in terms of cases and controls that we recruited, how long it took us. So 36 months overall to get to the 150 cases that we wanted in the original cohort. So, and these are just literally single blood sample or single saliva sample. We already had the clinical data in CPRD, but that's how long it took. And that can be the issue in terms of actually undertaking a precision medicine study. But fortunately, we have got there. We've been able to exome sequence these patients. I'm not going to show you the data. And we're identifying new variants which predispose to statin myopathy and so on. And that's coming along very nicely. So it was worthwhile in the end persevering with that particular study. So what are the other challenges? And I think those were alluded to earlier. Clearly, particularly with cancer, but also it's going to become important with other areas, is that you need access to diagnostic test investigations as part of your screening process. And clearly, for example, with cancer, what was talked about earlier, and I'm sure Eddie is going to talk about it, with PD1 inhibitors and so on. But also, when you are undertaking some of these biomarker stratified trials and the platform trials that James talked about, sometimes you do need technology within the center where you can actually start recruiting them, and we've been trying to do that using point of care technology sometimes, where you can actually democratize genomics, taking it to really district general hospitals, so you can start recruiting for precision medicine there, and you need that democratization of genomics to be able to do that with particular phenotypes you may be looking at. Clearly, there are also impacts for patients in taking part in precision medicine studies. If you screen 100 patients and 97 you're not going to be able to recruit, then 97 patients are going to be disappointed for not being able to take part in that study. And cancer, that might be a particular issue. So there's a need for clear information. I know that Focus 4, etc., are undertaking some very novel ways, developing DVDs, etc., to be able to make sure the patients understand why only 3 out of the 100 have been recruited. Because you have to scour the whole country, patients do have to travel greater distances, and clearly those are issues that need to be taken into account when you're designing your studies as well. Because of the complexity of some of these studies, I think we also need to think about the challenges of data capture, data management, and expertise at different sites. I told you about the skilled workforce. How much expertise will there be at local sites in terms of being able to phenotype the patients, recruit them, and so on, and bring them in? And again, I think that's very important as well. And clearly, The complex studies that we carry out now requires support in terms of CRF management and so on, multiple data points at different times, etc., spread over many weeks in relation to drug intake. So this is my area. So if I want to undertake particular studies for precision medicine, as a clinical pharmacologist, I may want to actually measure drug levels. We forget that in precision medicine there is a drug often in the middle of that. We may have all those omics technologies, so we need to forget the drug as well. So I actually measure drug levels at different time points. And I may want to do it at different time points in relation to drug intake on different days. And in order to do that, I really need CRFs to have the expertise to undertake that kind of studies, and sometimes it's lacking and so on, and I get very little uptake sometimes, so I have to do it locally. And that reduces the numbers I can recruit and so on. So there are challenges such as that. So obviously, particularly when you're looking at some of the rare phenomena, and I am particularly interested in serious adverse effects of drugs, and this slide shows you a patient with toxic epidermonecrosis. And this is where your skin blisters off. And this patient that I looked after at the Royal Liverpool Hospital had 60% of their body surface area blistered, and they had toxic epidermonecrosis after receiving an anti-epileptic drug. Fortunately, it's rare, but it's important for us to be able to study these rare diseases and rare adverse events like this. But the challenge is recruiting these kind of patients and identifying them and bringing them in. And so what we've been doing is working with a patient group called SJS Awareness UK, and they've got a fantastic network through social media, and they've been able to identify more patients for us than we've been able to identify through NHS services, which is very important. So this is where you can use patients to be able to help you recruit. But the problem, then there are other problems that occur if a patient actually helps you identify patients. So we may get a patient being referred to us, or patients contacting us, let's say from Cornwall, and so on. So it's not easy to get from Liverpool to Cornwall, by the way, so that's a challenge. So, but what we do is that, first of all, a patient's contacted, and this is a typical journey for recruiting one patient with a rare disease like that. Patient contacts us. We need to make sure that we've got all the governance approvals in place where they are. Again, that can take a while. We need to sometimes we ask for a local PI, but you don't actually need a local PI because we've already got we already got the patient saying that they've had this particular disease process. You have to check with the patient that they're healthy to taking part and so on in the study that we want to undertake, and then the nurse has to travel down to be able to assess the patient and be able to recruit them. But the patient was ill when they were having this particular adverse event, and they may not have all the clinical information. And so they give you a limited amount of information, but then you actually have to contact the clinician for information. And when we often contact the clinician, we don't get a response, so we can't actually get the information. So you patients very willing to take part. They may have pictures they've taken on their iPhone. We can see them that they've had those blistering conditions, but we can't actually get the clinical information. Then you actually go through the multiple clinicians they've been involved with, try to get the clinical information. Eventually you get some success, but it's taken you about 6 months to recruit one patient when you get down to the bottom, when you've got the sample from them. So those are challenges, particularly with rare diseases, rare adverse events, in terms of trying to recruit patients. So I think it is important we understand that there are huge challenges, but there are also fantastic opportunities as well. And I think there's great infrastructure that's been developed through the funders, but also through the CRN. Hopefully today the idea is that we can actually try to develop solutions to these challenges so that we can actually have a much more streamlined process, so we can try to recruit patients quickly into studies, time and target and so on. One of the issues for trying to recruit one patient like this over 6 months is that we often don't take into account the resource requirements. When we talk about costings and support costs and so on, we often do it on a very perhaps model which maybe needs to be maybe changed or enhanced, particularly for these kind of complex studies when you try to recruit one patient for a rare disease. So I'll stop there and go on to the next talk, but I think what I want to do is to get people to ask questions as well of the speakers in the next session. So has anybody got any questions for me before I move on to the next speaker?